So a good afternoon to all of you. This is Narottam from Shantiniketan Viswa-Bharati, and this is my. Hey, where is this? One more. Yeah. Okay. So the eight. Okay. Just a minute. Just stop. Ini pin ko ni. Relax na to. Okay. Yes. Now it is okay. So good afternoon to all of you. The topic on which I will discuss is that genetic regulatory switch between Sub1a and SK, the key adaptive mechanism behind shifting from quiescence to elongation in submerged rice. Now regarding the so first of all, I have to tell you that there is the background of the origin of my topic on which I am doing research here. So you all we know that is submergence tolerance is one of the major problem in lowland rice fields of the eastern India, particularly Assam, Odisha, and West Bengal. Now there are two well mechanisms, the two well described, well developed mechanisms. That is, we know that is one is quiescence and another is deep water flooding. Now, due to changes of the environmental condition, because you know there is a huge changes in the environmental condition, for which though these two mechanisms which we known as quiescent as well as the deep water flooding, there are two separate mechanisms is working. But theoretically, those that is available in theory, but in practical, what is happening due to this geopartition of this particular environment, a particular field have to face initially with this type of first class, then it is under the one type of water stagnation. So a single plant have to face both the things, that is both the first flood as well as this type of deep water fight. Sometimes it is one type of water logging. So it is very much difficult for a plant to become survive if a particular rice line have a single loci, either that is sub1 or that is the snorkel loci. Because you know sub1 in this particular picture it is showing there is two particular loci. One is that is the sub1 loci, another is SK loci. Two are interplaying into different condition. In the left hand panel, it is showing that is under the submerged condition, what is the how a particular plant face, and whenever the water level is up to 5 to 50 centimeter, then in generally that is the sub1 loci is interplaying. But whenever the water level goes more than 50, then this particular SK locus is interplaying. But the condition is that now the situation. So this is the one diagrammatic view of the genome of rice, particularly in chromosome number 9 and 12. Two loci are located, particularly two QTLs are located. One QTL is sub1 cuticle. It has sub1 A, B, C, 3 different components, and in chromosome 12 there is the snorkel loci, and it is showing that is the both of them are ethylene responsive factor. Whenever a particular plant undergo under the this type of oxygen deprivation, anoxic condition, then they are interplaying through the ethylene responsive factor. So this concept is well developed, and you know from the Odisha there is one of the rice land races which is commonly known as the FR39. The sub1 have been introduced into a number of other high yielding background to develop a number of high yielding rice lines like Swarna sub1, Subida sub1. So up to this everything is okay. So there is sub1 a good number of Swarna sub1 or this type of rice lines is available to the farmers. They are doing in this way for the lowland, particularly on the mid lowland rice ecosystem. But problem is that whenever a particular field is suffering from a water stagnation, that is initially a particular field is suffering from this type of fast flood, then the water remain around say 70 to 80 centimeter for a more than 1 month, then plant have to face, that is the then during that time sub-1 loci is not working. Then the situation will be that plant have to be die or producing very little amount of yield. So plant is not too much productive. So that is the reason these are the different type of fact what I have mentioned here. And the last paragraph I have mentioned, but the genomic diversity study on a number of lowland rice lines like this a number of rice lines I have mentioned, Bangasali, Kalirai, Sadajavda. Interesting, this type of rice lines contain both the sub-1 loci and SK loci. But most of the literature still now reported, they are mentioning that a particular rice genome may contain either sub-1 or SK. Both of them cannot be present at the same time. If they are present, generally SK remaining non-functional. But then the problem is that what will be the condition in case of those rice lines which I mentioned here, a very few limited lines which contains both the loci sub-1 and SK. So what will be their particularly expression profile as well as regulation, that is not known. That is the main aim of my topic, my talk today. So in these cases, I have collected, that is our group has collected 3 different sets, one from West Bengal, another from Odisha, another from Assam. So today I will discuss only for the Assam groups. So these are the different type of Assamese rice lines, particularly they face to face the first flood as well as deep water flood. So these are the different type of rice lines: Kalapatia, Bangasali, Samba Masuri, Khada, Kusma, Purnendu, Ranjit. So all these lines are not typical land races. There are some rice lines which are also improved rice lines. Say Purnendu is one type of such rice lines, Ranjit is one type of such rice lines. But most of them are actually in the different type of land races. We have collected around 25 such rice land races, of which 10 or 12 shows the good results. So I am showing only the 10 these things, only the 10 rice lands I am showing here. You see, this is so the key problem was, what is the most effective genetic combination of sub-1 and SK? Because sub-1 is a QTL, it has a several type of components. Sub-1 ABC, again sub-1 ABC, sub-1 1, similarly SK, SK1, SK2. So what is the actual or what is the good combination which is giving the best submergence tolerance, not only for the one type of plant, that is both quiescence as well as elongation strategy, under both quiescence elongation strategy, which will give the maximum amount of this type of submergence tolerance. You see, this is the different type of land races or some improved line also. And here the conventionally 2 different rice lines, one is used as a negative and another is positive check. That is IR42 is a negative check and FR13 is a positive check. Then all the different type of normal, what is the commonly used as physiological screening, anatomical screening, and the different type of that is the different type of biochemical screening and expression profile, all the things we have performed. So some of the results I am showing here, our group have recently this. So you see, the basic thing is that for this selection of those rice lines, we have thoroughly studied the availability of sub-1 SK locus and sub-1 locus and SK locus in all those rice lines. The right hand side is the panel showing it is the fingerprint profile with the sub-1 loci upper one, and the middle one is showing the SK locus. You see, out of these rice lines, all are showing the sub-1 locus, and SK locus, for SK locus it is showing the VB69, which is FR39. There is no report that is FR39 having the SK locus, because you know FR39 is famous for quiescence strategy, but it is showing it is also the SK locus. Then the problem arises, so what will be the mechanism of regulation? Because a particular genotype, it contains both sub-1 and SK locus, what will be their regulation? But interestingly, according to the literature, though in FR13 SK locus is present, it is not expressing. In our study also it is showing, it is totally showing one quiescence strategy, still, but it has some SK locus. Maybe SK locus in dormant form or something. That is the main theme of our study. That is what is the regulation of this one. how the gene expression profile regulates this particular two locus according to the different type of depth and different type of duration and depth of the water level. Now it is showing that is in the BB604 and 605. These are basically one. BB604 is one type of land bases. It is known as Gangasali. We have all the different type of rice, the different type of gene expression profile, mRNA profile, transcriptome profile. All we have used on this Gangasali variety because Gangasali is so because first one is Gangasali 604 and another is 605. There is the Kalakutia. Both of them shows SK locus. SK locus as well as Sub1a locus, but the physiological performance shows there is Gangasali is performing much better than the Kalakutia in terms of combination of both the two type of there is growth, there is quiescence strategy as well as elongation strategy. So in the lower panel, in the left hand side lower panel, it is showing that is the how the Sub1a and SK locus present in this Gangasali along with the that is the FR13 and IR42. In IR42, though that is the Sub1a locus is present, SK locus is not present, as literature is supporting this view. Then it is the you see different type of allele, different type of the right hand side it is showing the different type of Odisha and as well as in West Bengal rice lands, for which for those lines we have studied details about the locus, details about the different allelic form of Sub1a and SK. That is we have already published this thing, we have not presented here. Now regarding the physiological performance, in physiological performance it is showing there is both the IR42, FR13 and Gangasali, they shows a contrasting different particular physiological part. And what we have done, we have seen that is the shifting from the Sub1a and SK. There is how a plant grows quiescence from the quiescence when it shifted. That is from the quiescence to elongation. It has been shown that it is somehow done. It is somehow play within the 8 to 13 days. That is from the 8th day to 13th day. In between this period, this particular shifting happening. So between 8 to, because initially we have studied the 7 days, 14 days, 21 days post submergence period. So we have studied this one. Then in order, then we have noticed that is this shifting is basically taken in between 8th and 13 days, 14 days actually. So from 8 to 13 days, every day we have studied the physiological performance, the biochemical performance, the gene expression profile. Every day we have studied, and interestingly we have noticed that is yes, in case of Gangasali, that is which is showing both the elongation and both the quiescence as well as elongation, and this shifting is happening in the 12th day. So 12th day is our main priority. If we, because in we are our hypothesis is that there is the shifting from the Sub1 activity to SK activity, it is happening on the 12th day. So we have to study all the different type of tests, biochemical, physiological and expression profile during this period, that is the throughout this day. So in this it is showing Gangasali is showing a contrasting and very good results, and then it is then similarly all the different type of rescues, because we know that is that there are a good number of enzymes, particularly the which are involved in this type of anaerobic respiration. Alcohol dehydrogenase, sucrose synthesis responsible for sugar locating, then pyruvate decarboxylase, all the different type of enzymes which are commonly reported to be linked with this type of submergence tolerance. For all this type of enzyme, we have performed the real-time PCR as well as the amount of this particular enzyme activity. All that we have studied, and we got a good results. There is a contrasting results. There is maybe in the 12th as well as 13th, in between the 12th and 13th. In this period, the shifting is happening. Though in all the cases they are not showing the same pattern of result, there are some deviation. But for as a whole, it has been shown that I am not going into details about this one. But it is showing that is the graphically it is showing that is the they basically whenever we compare with the control plant with the test plant, it is showing that particular time period, particular point, they are shifting from one phase to another phase. Then this is the profile you see, that is the particularly expression profile of the Sub1 and SK locus, and there is the Sub1 and there is the all the Sub1 and SK locus throughout all the days. It has been studied, and it has been shown that yes, the physiological biochemical, which in both the study what it was being displayed, that is the particularly shifting from the quiescence to elongation. On the 12 days, somehow the plant is shifting from quiescence to elongation periods. Then this is then after that one, because regarding the switching as well as regulation of gene, there is another candidate genetic elements, that is microRNA, because microRNA are also responsible for different this type of, that is the fine tuning of the different loci, because microRNA can target a particular loci, then accordingly they have controlled their expression. Sometimes it is sometimes it is just block a particular loci so that it will not be expressed. So, and till now there is no such mRNA available for this particular Sub1 and SK. So we have initially we started one bioinformatics analysis to find out what are the different type of microRNA loci available in the RISE database, which are targeting the Sub1 and SK locus. Then those microRNA we have studied those microRNA. So here it is the what I told you, that is the same thing it is mentioned here. Then it is actually the pipeline. That is, initially we have to isolate the particularly those mRNA. Then those mRNA we have to find out whether those mRNA binding in which location. Then out of those location which is thermodynamically constant, the thermodynamically favorable, that means which is the best candidate microRNA for that particular Sub1 and SK locus. Here I am showing only the SK locus because this data has been already published, but Sub1 loci is not yet published, so I am not presenting here. So we have identified this, the Osamir 319P and Osamir 439. These are the two candidate mRNA which are supposed to be targeting the SK locus. Then in the next phase, the same thing we have studied, that is their sequence diversity, how much they are conserved, and their interaction with the particularly the SK, that is the SK locus. And here are the molecular network, that is what are the different type of mRNA supposed to be interact with the particularly SK locus, because SK locus is two form, one is SK1 and SK2. All the different type of microRNA, they are network analysis, this one, and the lower one is, that is the bioinformatics analysis. It is showing, because whenever a particular microRNA targeting a particular target, then where the binding is most bioinformatically suitable or more economic, so that will be the best mRNA, and which is targeting the particular target loci. Then finally we have to validate our concept, our hypothesis. Then what we have seen, whenever we study at the stress material, that is the SK locus, its function, its expression, along with the mRNA. Here mRNA 1319B, this is one mRNA we have studied. Another mRNA did not show very good result during the validation study. So here MIR 131B, it is showing the good result. So it is showing that is whenever the mRNA expression is increasing, mRNA level is decreasing. So that is what we want to study, want to prove. That is already proved for the one mRNA. From here we can say that this is the mRNA which may be the responsible for this Sub1 loci fine regulating. In the second job, what we will do, that is what are the total mRNA which are responsible for both Sub1 and SK at a time, and what are the different type of that is the same thing we will do, that is from 8 to 13 days, on particular which day, that is all the microRNA is regulating these things, as a result of which this type of regulation, this type of switching from Sub1 to SK locus will be done. So, and here are some of the relevant publications we have done for the last 5 or 6 years. SK, there is for SK locus, this one study of physiological responses with L diversity of Sub1 and SK, where it is showing the total diversity, and the lower one in the right hand sides. This paper is yet we have there is communicated, is under review. That is how the Sub1 and SK is interplaying. There is the shifting. Okay, and in the left hand side, in the below, you see mRNA regulation of Staphylococcus in rice under submergence. So these are the different type of publication we have done in the last 5 or 6 years. So I am finished. Thank you for providing me this platform to present my talk in the online. Thank you all. I will be happy I able to accept some questions.